These are some sample cell models you can make for your project. Most of them use a styrofoam ball, but you don't have to. This one didn't. So I'll go over the parts that they show on these real quick. This has really been one of my favorite ones here. Let's see what some of the parts are. Well, of course, we have the nucleus, and then we have a nucleolus here. You can see the DNA strands they made with strings here. And then you can see the nuclear pores here and the nuclear envelope surrounding the nucleus. All of this, of course, styrofoam is the cytoplasm. And of course, we have the cell membrane, the outside, also called the plasma membrane. Some of the organelles, well, the mitochondria here. Here, I like how they made the squigglies on that to show the matrix, the cristae inside the mitochondria. Then we have the ribosomes, these white pens, which are on this pipe cleaner blue, which represents the endoplasmic reticulum, the roadways. So since the ribosomes are on this blue part here, that would be the rough ER, endoplasmic reticulum. This would be the smooth ER. It's still endoplasmic reticulum, but no ribosomes on it. The Golgi apparatus is similar to the ER, except it doesn't have ribosomes, and it's a different color. And that's for storage instead of movement of substances. Then we have some vesicles, which would be coming off of the ER. And let's see if they show any vesicles. I don't see, here's a vesicle in green, because remember, vesicles have to be the same color as the Golgi or the ER, because they're going to branch off of those. Now the yellow, since it's a different color than the ER or Golgi, would be the lysosomes here. These fibers here would be the cytoskeleton fibers, the black. So that leaves us with the centrioles, which look like stacks of dynamite, usually 2 of them. Let's move to another one. Here's another one. And we won't see all the organelles on here, but I want your model to show all the organelles. So here we have nucleus, nuclear pores, nucleolus, nuclear envelope, cell membrane, cytoplasm. We see the cytoskeleton fibers. We see the mitochondria here and here. This would be the ER and the ER here. That looks like rough ER with ribosomes, smooth ER. Different color here, so that's the Golgi. Vesicles would be in blue, so that means the lysosomes would be the yellow. They might have made these vacuoles, which we didn't cover. And then this would be the centriole here, and the cytoskeleton fibers. Here's another one. Now I like this one too. This shows some little cilia on the outside of the cell membrane. See? And then we show the cell membrane nicely there, the pink cytoplasm. Here's your blue nucleus with the nucleolus, nuclear envelope or membrane. Looks like this is the ER here. They showed some ribosomes on this ER, making that the rough ER, and that's a smooth ER. Here's a different color Golgi here. Mitochondria with the squiggly insides making energy. Those would be your centrioles there. These would be vesicles because they're coming off the Golgi. And these, purple, would be the lysosomes in this case. See some rough cytoskeleton in there. Here's another one. Nucleus nucleolus. ER with ribosomes. Smooth ER over here. Here's your Golgi, different color. Looks like this would be your vesicles in blue. Here's some of your cytoskeleton. Lysosomes in yellow. Looks like this I would call, having trouble distinguishing between the mitochondria and the centrioles. Let's see what they tried to do here. I would say this would be the centrioles, because there's two of them. But usually there's a whole bunch of mitochondria. It looks like they tried to do a little maze inside there. These could be vacuoles or peroxisomes as opposed to lysosomes. But the vesicles, remember, would have to be the same color as the ER or the Golgi. So in this case, the blue. And lastly, we had a little model here. Nucleus, nucleolus, DNA, nuclear envelope, ER with ribosomes, Golgi, mitochondria. This looks like a vesicle here and here. Also those. And then you have your centriole and cytoskeleton. Now it would be hard to ask which one was a lysosome on this, unless they're making it these. But this, it looks like the ER in blue and the Golgi in green. So we could say that the orange or yellow is the lysosomes, and these would be the vesicles. But be original. You don't have to use a styrofoam ball, although you can get them conveniently in an art store. You can do a flat model. Doesn't matter. This one's been the best one I've seen so far. See if you can top it. Now, use anything you want, except don't use any edibles. No edible parts for the models. I found that out the hard way because I stored some in my basement once that had used little noodles and jelly beans. And I went down there once and there was mice droppings in my basement. And some of the jelly beans had been eaten. So no edibles. Be original. And to get credit, I have to be able to use it. Don't just slap it together. Make it accurate so all the cell parts we went over. All right, that's it.